It's my great pleasure to introduce Dr. Sutapa Roy. She works in IIT Gandhinagar in the physics department, or physics discipline as they call it. She's a soft matter physicist. She did her PhD from JNC ASR, and she was a postdoc in several places in Germany, including Stuttgart, especially Stuttgart. And she joined IIT Gandhinagar in December last year, and she's been working. She's an assistant professor there, and she's going to speak on the dynamics of fluids around laser-heated colloids. And due to various glitches, she'll be speaking from my window. But this is Sutapa.
Okay. Yeah. So you hear me, right?
Yes, we can hear you.
Okay, good. Yeah. So let me start by thanking Professor Nilima Gupta and Professor Shayan Gupta for giving me this wonderful opportunity to present some of the works. So I really apologize for the delay, but I hope we can finish in time. So so what I'm going to talk about is dynamics of fluids around some laser-heated colloidal particles. And okay, so here's a brief outline of the talk. So as you see that I'm going to have in the talk some colloidal particles, and these colloidal particles are basically going to be laser heated, and they're kept in a fluid. And then we are going to look at the dynamics of the system. So the basic outline is the following. I'll first introduce to you what do I mean by these laser heated colloidal particles, and what is the basic question that we are addressing in this work. I'll explain next the model and the methodologies which I work. So this work is, so by training, I am a theoretician. So the work which I'm going to present is mainly going to be based on the theoretical work. However, some of the results that I'll show are also from the results, our collaboration with experimental colleagues as well. So followed by that, I'll explain some of the results and finally the conclusion. So, okay. So to give you the brief idea, what we are looking at here is the following. So we have some spherical colloidal particles, and these colloidal particles, we put them in confinement. So we have two glass slabs, which are kept parallel to each other. And in between these glass slabs, we have a fluid. And there, as you see here, we have the colloidal particles, which are kept confined. So you have this setup, and then you shine laser on top of this fluid. Then what you see here is basically under certain condition, this colloidal particle that you look at, that undergoes some kind of active motion, basically. And these particles are made in such a way that if you look at the right and the left hemispheres of the particles, they have different surface properties. And in the scientific terminology used for that is Janus particles. So these particles, if you look at the right and the left hemispheres, they have different surface properties, because of which they are called Janus particles. And these particles then, when you heat up, they undergo some kind of active motion. And we see the formation of some asymmetric structure around the particle, like you see in this schematic picture here. So if you look at these colored layers here, different blue and the yellow colored layers, those are basically some patterns which are forming around the Janus colloid. And the question is, of course, what is the mechanism behind the active motion of these Janus colloids? Okay? So that is one thing which you are trying to address. The next thing is, if you look at the structure which is forming around the particles, they are clearly asymmetric. Okay? And the question is, why are they so asymmetric? In the sense that, are they asymmetric only because you have a Janus particle which have different surface properties, or they are asymmetric because of something else as well? So that is one thing which we will try to address in this talk. And the third thing is, of course, why is it important at all from a broader perspective? So these are the three questions which I'll try to address in the next couple of minutes. Now, before going into that, let me spend one slide basically, which I thought I will keep here in terms of the students. So if you look at this the system here, which I'm talking about, it's clearly undergoing some kind of phase transition. Okay, so for example, these patterns which you are seeing here, this yellow color and the blue color patterns, they are forming because the system is having some kind of phase separation, which is related to some kind of phase transition. So I have one slide here, which are mainly meant for the students trying to understand what do we mean by phase transition and phases. So phases, like we know, we came across during the undergrad days as well. So matter exists in different phases, solid, liquid, and vapor. And that's what is being shown here in this cartoon-like picture here, where you see basically all the three phases of water. So you see the solid phase, which is the ice, the liquid phase, which is the liquid water, and the vapor phase, which you don't see here, of course. And whenever there is a transition from one phase to the other, that's called phase transition. So in the second picture here, I show a schematic picture of a normal chemical system in the pressure temperature plane. So the curved solid lines that you see, they basically mark the boundaries among different phases of the system, namely the solid, liquid, and vapor. So if you look at the green curve here, that marks the coexistence of the solid and the liquid phases. If you look at the black curve, that corresponds to the coexistence of solid and vapor phases. And similarly, if you look at the blue curve here, that marks the coexistence between liquid and vapor phases. Now, the point to note here is that if you look at this green curve here, which is known as the melting curve, which is the coexistence between solid and liquid phases, this curve continues forever. Whereas, if you look at the blue curve, namely the liquid gas coexistence curve, that terminates at a certain point, which is known as the critical point. Okay? So if you look at this black dot here, TCPC, so that basically marks the critical point. And the idea is that if you have a system which is at a temperature higher than this particular temperature, or at a point beyond this critical temperature, there is no difference between the difference intensity between the liquid and gas phases of this system. So one can always go from the liquid phase to the gas phase without encountering any jump in the density. And this density, in the terminology of statistical mechanics, is also called the order parameter in this particular case. So in this case, you can go from the liquid phase to the gas phase without encountering any jump in the order parameter. And that's why this kind of transition, which you see here, it's called a continuous phase transition or a second order phase transition. So now if you go back to the system which I am interested in, I have some colloidal particles which are put in a fluid, and that fluid is close to a continuous phase transition like this. Okay? So we are going to deal with the fluid which is close to a continuous phase transition or a critical point. And then within that fluid, I'm going to suspend colloidal particles. And okay, so just 1 minute. Yeah. So I'm going to suspend these colloidal particles, and then we are going to heat up these colloidal particles. And the question is, what happens next? So in this picture here, if you look at the first picture, what I show here is basically the phase diagram of this solvent in the temperature concentration plane. Now, instead of a single component liquid like water, in this case, we have a binary liquid, which means that we have two liquids which are kept mixed with each other. Okay? So that's why the term binary comes into the picture. And by concentration, I mean the relative number of particles of one of the species. Okay? So if we look at this first picture here, y-axis is temperature, the x-axis is the concentration of one of the phases. The red point, if you look at it, that's called the critical point. The same critical point which we were talking about in the last slide. Now, see here. This critical temperature, if you look at it, it's actually close to the room temperature, 34 degrees centigrade. Below this critical temperature, the system is in a homogeneously mixed phase, which means that if I look at the binary fluid, there's no way I can distinguish between the different phases. They are homogeneously mixed. Okay? Then if you heat up this liquid, and the liquid temperature increases, and it enters into this gray colored region here, which is also called the two-phase region, there basically the two different phases of the binary fluid can coexist with each other. So now imagine a situation that you have this homogeneous fluid which is in equilibrium at a room temperature. Okay, so everything is homogeneously mixed. Now you suddenly increase the temperature of the fluid, and you do it suddenly. Then clearly the system has now got it into the two-phase coexistence region, and it has not reached the new equilibrium state yet. So it will try to go from the old equilibrium state to the new one, following a non-equilibrium dynamics, and that non-equilibrium dynamics is known as the phase separation dynamics. Okay? So this is what happens for the fluid which is under consideration. Now, once again, if we go back to the experimental system, which I was mentioning before, we have these glass slabs, and we have the fluid in between, and we have the colloidal particles which are suspended in the fluid. And then you are turning on the laser. Now, the laser intensity is chosen in such a way that only one half of the colloidal particle, so in this picture, the yellow half here, the gold part, that absorbs the light, so that gets heated up, whereas the left half, which is the blue half, that does not absorb the light. Okay? Similarly, the fluid, if you look at it, the fluid does not absorb the light. So the wavelength is chosen in such a way. So this clearly gives rise to the formation of some kind of temperature gradient in the system. Because you see, the right hemisphere of the colloid, which is the gold cap, that absorbs the light, it gets heated up, and that is actually going to propagate from the colloid into the fluid. So depending on the proximity towards the colloid, the fluid is going to have different temperatures at different positions. So you clearly have an inhomogeneous temperature field which is set up into the fluid. And this field, if you think about it, it's actually time dependent. Okay? And this time dependent temperature field gives rise to the formation of this asymmetric structure, which you see in this second picture here. So if you remember in the introduction slide, I told you that what's the basic mechanism behind the formation of this, the local structure formation. So it's clearly related to two things. One is the time-dependent temperature gradient, and the second thing is surface effect. So these colloids are prepared in such a way, typically, they have a surface affinity to one of the two components of the fluid. So they typically prefer one of the two components. So for example, say for the fluid, if I have water as one component, it's either going to like water or it's going to dislike water. So depending on that, you are going to have different kind of structures which are forming around the particle. So this is the main experimental setup, the thing which is our experimental colleagues observed, and we wanted to find the basically the mechanism behind it. And for that, we did a theoretical calculation, mainly based on mesoscopic theory and some kind of numerical calculations. So to give you the analogy between the experimental experiments and the theoretical modeling. So in the experiments, what you do is that you heat up the colloidal particles, and then you see the structure which is forming around the particle. Okay. Now in theory, basically what we do is that we actually cool down the particle. So starting from a high temperature, we are actually cooling down the colloidal particles. Okay. And the surface properties, which I was talking about, hydrophobicity or hydrophilicity, that is taken care of in theory by appropriate symmetry breaking surface field and some kind of universality classes, which I will not get into the details. But if you're interested, we can come back to that later. So the basic idea for the modeling is the following, that we have whatever non-equilibrium dynamics is going on, it's happening under the presence of two fields, one temperature field and one concentration field. These two fields are time dependent and they are coupled to each other. And we are going to study the time dependence of these two fields under certain boundary conditions. And we are going to consider appropriate weighting properties as well. Okay, so here is the basic modeling from the theoretical side. So as I told you, we have two fields. One is the temperature field, another is the concentration field. So the time dependence, so the time evolution of the temperature field is dictated by the heat diffusion equation, which you see within this yellow color box here. So capital T is the temperature field, and small t is time. And the capital D, if you look at this equation on the right-hand side, that's related to some diffusion constant. So maybe let's not get into the detail, just to tell you that we're actually trying to solve this heat diffusion equation, which will give you the time evolution of the temperature field. Now coming to the concentration field, so that is basically taken care of by appropriate something known as the Cahn-Hilliard equation, which comes from the Ginzburg-Landau free energy functional. So the idea is the following, that if you look at the free energy of this system, you can write it as a combination of some square order parameter square and some square gradient terms, like you see in this equation here. And then this fluid, if you look at it, if you look at the total order parameter, that is actually conserved. So this conservation gives rise to some kind of concentration current into the system following the continuity equation. And this current you can write in terms of the gradient of the chemical potential. So the J here is the concentration current and mu is the chemical potential, okay, which is basically the gradient of the chemical potential, and capital M is the mobility. So if you do a few steps of algebra, you can actually show that del psi del T, which is the time evolution of the concentration field, you can write it in terms of all the terms which are appearing in the Ginzburg-Landau free energy function. So, and then if you do this some basic steps, you basically arrive at this equation, which is written in the yellow box here. Now, the thing to note here is that this kind of Cahn-Hilliard approach, if you look at the theory, that is mainly done for isothermal systems, where the temperature is the same everywhere in the fluid. But in this particular case, we have temperature inhomogeneity. Okay? So we have to take that into account, and to do that, basically, the way we did as a first approximation is, if you look at the first term here in this yellow colored equation, we have a coupling between the temperature field and the concentration field. So capital T and psi, they are actually appearing as coupled with each other as the product terms. So that basically takes care of the temperature inhomogeneity and the coupling between the temperature and the concentration field. So the idea is the following, that you start with a certain initial configuration, which is the homogeneously mixed system. And then you solve the time, the heat diffusion equation and this Cahn-Hilliard, modified Cahn-Hilliard equation, and they will give you information about the time dependence of this temperature and the concentration fields. So that's what we do, and let me show you some of the results. So on the left-hand side picture, I show you the snapshot of this fluid around the heated colloidal particle. So the gray part here corresponds to the colloid, which we basically heat up, and then we have this structure which is forming around the particle. So first of all, what you see here is that you have a surface adsorption layer which is forming on top of the colloidal particle. As time progresses, you see this surface layer is actually getting thicker and thicker. And away from the colloid, you have some kind of surface-directed spinodal-like patterns which are forming, which actually get bigger and bigger as time progresses, until at a very late time, t equal to 1000, in the steady state, you have only one thin spherical shell which is formed on it. So that's basically corresponding to the steady state. Now one can also quantify this kind of non-equilibrium dynamics in terms of something called the two-point correlation function. So in the second picture here, that's what is shown. Y-axis is the correlation function and x-axis is this the distance from the colloid. Different colors here correspond to different times. Okay? So the black color corresponds to very early time and the yellow color to a very late time. Now, if you look at the leftmost part of the x-axis, so when the x-axis is equal to 1, that corresponds to the colloid surface. Okay? And as you go towards the right, you are actually moving further away from the colloid. So first of all, to note here is that close to the colloidal particle, you have a surface adsorption layer, which is corresponding to this positive value of the correlation. Next to that, you have a depletion layer, okay, where the correlation is actually negative. And then depending on time, for example, at red times, if you look at the red data, you see you have one more layer of fluid layer around the colloid and one more depletion layer. So basically, as time progresses, you have more and more layers which are forming around the colloid, and that leads to basically multiple peaks in the correlation function which you look at. So these results are mainly for systems when you have basically they are homogeneously mixed, which means that both the sides of the colloid, they are preferring same type of particle. So in this picture, let's now move on to the Janus colloid. Okay, so in this case, we have this Janus particle. Once again, you take the particle, put it in the homogeneously mixed fluid, and then you turn on the laser. Now, the thing to note here is that if you look at from t equal to 0 to t equal to 400, you have not turned on the laser at all. So whatever is happening from t equal to 0 to 400 is happening entirely because of the surface enrichment phenomenon. Okay? So because I told you that these colloidal particles have a preferential attraction to one of the two components of the fluid, that generically they try to attract that particular species, and there is a surface enrichment layer forms which compose of that species. So that's what you see here, starting from t equal to 0, the homogeneous mixed phase, you have a surface absorption layer which is forming and which gets thicker and thicker, and finally in the steady state at t equal to 400, you have a thick surface absorption layer which is formed. Now at t equal to 401, you turn on the laser. So the pattern that you see here at t equal to 400 has now formed in the presence of a temperature gradient. Okay? So then if you compare these two patterns at t equal to 400 and 1400, you clearly see a huge asymmetry at t equal to 1400. And this asymmetry has clearly been generated due to the presence of this temperature gradient. Okay? So the conclusion is that when you have a temperature gradient in the system, because of something known as the Soray effect, it leads to strong enhancement of asymmetric enhancement of the concentration of the fluid. And that is giving rise to this kind of asymmetric structure. So if you remember in the introductory slide, I mentioned the question that this asymmetry that you see around the Janus particles, is it entirely because of the Janus nature of the particle, or there is something else? And the answer is that there is clearly something else, and that something else is the temperature gradient. So the stronger the temperature gradient, the higher is going to be this concentration asymmetry. And this has huge implications in the active motion of this particle, because the stronger this asymmetry, the higher is going to be the velocity of this active particle. Okay, so these are some of the results which were actually results from our numerical simulation. So after we did that, we had some kind of results from the experimental collaborations as well. I will not get into the details of that, but just to tell you that we had some nice, similar observations from the experimental side as well. So maybe I'll skip it in the interest of time. If you're interested, I can come back to it later. Let me now show you some results for confinement. So these colloids which I was showing you before, they are actually kept in a way that they are a bit something happened to the slide anyway. So those colloids were basically put in between two glass slabs. And the question is that depending on the separation distance between these two glass slabs, how is it going to affect the dynamics? So on the left side here, these results are presented for a thin film where this colloid is almost touching the two slabs. On the right-hand side, you have it for a thick film where you clearly see you have small space. Now, if you compare these two different dynamics which is going on, first of all, you have surface enrichment layers which are forming on top of the colloidal particles and on the glass slabs as well. As time progresses, these layers get thicker and thicker, and until you get the steady state pattern. Okay? So the basic non-equilibrium dynamics is the same in both the cases, both thin film and thick film. However, what is interesting here is that if you look at the left-hand side picture for the thin film, if you look at t equal to 100, you see I already have a fluid, a solvent layer formed, which is capturing the colloidal particle and the two glass slabs entirely, which means that it has now formed some kind of liquid bridge around the colloidal particle. If you compare it with the right-hand side picture at equal to 500, I clearly don't see any kind of strong bridge formation. Okay? And why is this important? Because it has application in colloidal assembly. For example, if you look at the left-hand side picture here at equal to 100, if I now put another colloidal particle next to this colloid, simply because of this capillary attraction, which will happen because of this liquid bridge, the second colloid will then get attracted to the first one and they will form some kind of colloidal assembly. which will not happen in case of the thick film, because I don't have any kind of liquid bridge which is formed around the colloidal particle. So basically the confinement effect, how thick or thin your film is, that will have effect in the colloidal assembly formation. Okay. So these were some of the results which I was showing you for the basically for the fluid which is forming undergoing this non-equilibrium dynamics. Let me now show you some results for the active motion of these colloids. Okay. So for that, what I plot here is the velocity of these colloidal particles versus the temperature gradient. The same thing is plotted on both the pictures, left and right hand side. The only difference is that the left hand side picture is basically a philic colloid and the right hand side is a phoretic. Now, first of all, you see here, if you look at the rightmost part of the x-axis, that corresponds to a temperature gradient 0. So I don't have any temperature gradient at all. And then as you move towards the negative x-axis, I'm increasing the temperature gradient. And you see, as the temperature gradient is increasing, the velocity of the particle is also increasing. And that is something which you can expect intuitively as well, because if you look at the basic mechanism behind the motion of these particles, what happens is that you have this concentration asymmetry around the particles. Okay? So that is some kind of pressure difference. So these solvent molecules, they then try to go from the high pressure region to the low pressure region, and the colloid will then try to move in the opposite direction. So that's the reason why these particles move. It's basically called a phoretic motion. And clearly, if you have stronger temperature gradient, this asymmetry in the concentration is going to be higher. So the velocity of the particle is also going to be higher. And that's what you see in both left and right hand side pictures. What is interesting here is that if you look at the right hand side picture, you see, starting from temperature gradient 0, as you are increasing the temperature gradient, velocity is increasing, which is expected. However, beyond a certain temperature gradient, the velocity actually changes its sign. So from negative value, it then crosses over to a positive value. And what it means is the following. Initially, if the particle was moving in the direction of the cap, so in the direction of the gold cap, now it suddenly changes its direction and it starts to move in the opposite direction. And the question is, of course, why? So basically, same result was obtained by our experimental colleagues as well. And we are still trying to understand what is the physical reason behind this kind of change in direction of the particle. And some intuitive idea which we have is the following. So basically, it's related to the temperature gradient which forms around the colloidal particle. So in this picture here, what I show you is the temperature profile around the Janus colloid for different temperature gradients. The green part here corresponds to the temperature which is exactly at the critical temperature. The blue part corresponds to temperature which is higher, and the red part to temperature which is lower than the critical temperature. And as you see, in the leftmost picture, only a thin portion of the colloid is at a temperature which is higher. As you increase the temperature gradient, this blue part is getting bigger and bigger until and unless finally for a very large temperature gradient, you see almost all the, basically almost full colloid has now been at a temperature which is higher than the critical temperature. Okay? And this critical temperature gradient is intrinsically related to the concentration which is forming around the particle and to the concentration gradient. So what we guess is that, so this difference in the temperature gradient, the isotherm, that is giving rise to different concentration field around the particle, which is giving rise to this change in the direction of the colloidal particle. So with that, I will conclude the talk. So basically what we started is that we have some Janus colloidal particles which are kept confined in between two glass slabs. They are suspended in a fluid. The fluid is close to a phase transition. You heat up these colloidal particles, and then you see some kind of non-equilibrium dynamics which is happening around these colloidal particles. We study the mechanism of this non-equilibrium dynamics. You see the particle undergoes some kind of active motion. We try to understand what is the mechanism behind this active motion. Why does the particle move at all? And then we try to quantify the properties of this motion. And if you can keep these particles confined in a thin film, you see, depending on the thickness of the film, you can see the formation of some liquid bridge around the colloidal particle, which has application in the formation of colloidal assembly. So with that, I would like to thank you. And yeah, so yeah, take the questions. So, Acha. So I can stop sharing. We can stop the sharing.
You just ask them first. Can you stop sharing? Or does anybody want to say anything on any slide?
So shall we stop the screen share?
I mean, is there any questions? Maybe Anubhav?
Yeah. Sutapa, can you hear me?
Yeah.
Hi. So
I
I just wanted to check when you talk about the effect of confinement, how is the confinement actually incorporated into the model?
So that's taken care of by the boundary conditions. So basically, you have, if you look at the free energy of the system,
Right.
you have a bulk component, and because of the presence of the surfaces, now you have a surface component as well. And then you can write some kind of boundary condition, which is imposed on this free energy. And within that boundary condition, you can actually, take care of basically the surface effects. Certain surface field which you can include into the boundary condition, and the surface field will take care of this surface properties.
So will this boundary condition on the free energy be capable of actually incorporating the effects of the boundary in terms of the hydrodynamics, like how the colloidal particle, as it tries to move through this small gap, it would experience a very strong near field lubrication kind of effect. So is that something, or it's a different point altogether how it's addressed in the free energy boundary condition?
So we haven't yet incorporated the lubrication effect. So there are studies on similar kind of systems where they have done it actually. And yeah, so till now we have not taken that into account. But you're right.
Otherwise, yeah. But otherwise, when you right now the way it's being done, it's done via the boundary condition on the free energy itself directly.
Right.
And I'm
Yes.
guessing that it can obviously also account for when you have multiple colloidal particles, like particle-particle interactions. Even this can capture that too, right?
Sure. Yeah. Absolutely.
Okay. Thank you.
I think Saurav has a question.
Yeah, yes, ma'am. Yeah. What are the applications of this type of study? I want to know.
Okay, so these studies are actually motivated by some active matter systems, basically. Yeah, so basically these colloidal particles which you look at, so as I told you that they undergo some kind of active motion, right? So those are called the active matter systems. And one good application is that in terms of the drug delivery. For example, in case of the cancer treatment, etc., you often need to deliver the drugs in the body in a certain controlled fashion. And the idea is that you have to deliver them in such a way that it goes to the exact targeted cell and it does not damage the other cells, etc. So that's another targeted drug delivery, basically. So these have this colloidal particle motion, which you look at. You can get certain idea about the motion of these active particles, and they have rather applications in such more application-oriented fields.
Okay. Okay. Thank you.
Anyone else has any questions? Okay, I think no one has any questions. Thank you very much, Sutapa, for the nice talk. And
Thank you.
yeah, sorry for the issues related to the logging in and all that stuff.
Oh, no, I'm sorry for that. So it happened actually because I didn't bring my laptop and then there were some issues. But yeah, I think we'll manage. So,
Okay, then shall we close this meeting? Thank you very much. And
thank
it
you.
was a pleasure to have you here.
Thank you.
Thank you.